Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Syahmi Ahmad Mirosni and my number matric is S58738. Today I want to present my proposal, synthesis of silver nanoparticle and GNPS for Mariposa Christia vespertilinis. The first thing is introduction. Nanomaterial. Scientists agree that nanomaterials are characterized by the characteristic of a tiny cells that were measured in nanometers. Nanometer is a million of a millimeter that were approached at 1,100 times smaller than the diameter of human hair. When nanomaterial is a particulate from the particle can present either single particle or a agglomerator or aggregate nanomaterial. Although some spread that agglomerator or aggregate nanomaterial have a dimension beyond 100 nanometer are not nanomaterials, but it still retains some specific psychochemical properties characteristic of nanomaterials due to the large specific surface area, or also known as SSA. Nanomaterials have characteristics that make it distinct from others. One of it is that it has material from 0D, 1D, 2D or 3D that depends on the overall shape. The researchers found the importance of the nanoparticles when the researchers found that the size can influence the physiochemical property of a substance like optical properties. For example, 20 nanometer gold AU, platinum and silver have a wine red color, yellowish grey and black color respectively. These nanoparticles have distinct colors and characteristics as well as different shapes. Is 1.2 problem statement. There are two types of synthesis that can be done to synthesize GNPS. The first method is by green synthesis method, while the other one is by chemical synthesis. The green synthesis method is a method that is simple, eco-friendly, and it is also biocompatible. The chemical synthesis, on the other hand, is the experimental condition of an experiment that will control the size distribution of nanoparticle, its morphology, and the stability of the nanoparticle that all will affect the kinetic of interaction of metal, ions, and the reducing molecules. Next is 1.3 significance of study. The method that will be used in this study is the green synthesis method. There are a few reasons why green synthesis method was used. Firstly, because of the method is simple, it has made the green synthesis is eco-friendly and make it biocompatible. Secondly, the green synthesis should be done to reduce pollution, using less oil, using recycled materials, unlike chemical synthesis that were very harmful to the environment. 1.4 research objective. The objective of the study are to synthesize silver nanoparticle GNPS using the plant extract of the Mariposa Christia vespertilinis. Next, to characterize the physical and chemical properties of GNPS nanoparticles using Fourier transform infrared spectroscopy (FTIR) and the scanning electron microscope (SEM). The last one is to investigate the toxicity effect that were induced by the silver nanoparticle and GNPS. So next we are going to the chapter 2. 2.1 Silver nanoparticle GNPS. Based on a study, silver nanoparticle GNPS have about 20 to up 50,000 silver atoms that their diameters are usually smaller than 100 nanometer. GNPS usually have a remarkable antimicrobial activity even at a low concentration. This makes GNPS one of the most used nanoparticles in many fields. GNPS have been widely used in many fields, that is including the healthcare, medical, and the consumer field, and GNPS also has been used as an industrial purpose. GNPS has been in use various fields because they have unique physical and their chemical properties. The chemical properties of the GNPS are including the high electrical conductivity, the biological properties, and the thermal properties that can be found in GNPS. The chemical property of silver nanoparticles GNPS can be seen in the table 2.1. while the physical and the thermal properties of silver nanoparticles are listed in the table 2.2 below as include the figure 2.1 of the solution of silver nanoparticles. 0.11 Green synthesis AgNPS. Green synthesis is a better method to synthesize AgNPS. This is because the conventional physical and the chemical method to synthesize AgNPS is very expensive and hazardous. This is very essential to reduce to the various chemicals that were used in physical and chemical processes that result in the environmental toxicity. The green synthesis can have carried by us using fungi, algae, bacteria, seed, and among other things. For the green synthesis of AgNPS, we can use almost any section of the plants like the seeds, stem, and the leaf. There are also few secondary metabolites that were present in the plant like flavonoids, phenolics, polysaccharides, and terpenoids. This is for the reduction of the metals ion in the bulk metal preparation nanoparticle to obtain nanomolecules in response of the redox reaction. It is for the reduction of Ag to Ag0 that were achieved by the biological species during most green silver nanostructure synthesis process. The option for using green synthesis of AgNPS is by using solvent medium that were preferably water, an environmentally friendly reducing agent, and a non-toxic material for the stabilization of nanoparticles are three key concepts for preparing nanoparticles by using green synthesis. Natural reduction substance present to minimize the use of external agent. There are also basic requirements for green AgNPS synthesis, which is we need silver nitrate and a natural reducing agent. 2.2 Christia verpetolainis. Mariposa Christia verpetolainis, or commonly known as mariposa because this plant has similarity with the color and shape of the butterfly, is a plant that can easily be found in many parts in Asia, and it can reach the height up to about 1 meters long. Mariposa is an hibiscus plant and it lives in the terrestrial area. The characteristic of mariposa is its leaves are a compound with three leaves with the green color with stripes. Example of the mariposa Christia is below. 2.21 Phytochemical study of Christia verpetolainis. Because of mariposa have many potential to cure disease, there has been many study of the phytochemical of mariposa. The phytochemical of Christia verpetolainis are known in the research field to its medicinal properties. The recent study of Christia verpetolainis have revealed that the medicinal property of the leaf extract of the Christia verpetolainis plant, because it has phenolic content that were derived being screened for the antioxidant, anti-diabetic, and the toxicity properties by the following calculator method. 2.22 Total phenolic content. Phenolic compounds are an important plant constituent with the redox properties that has been associated with antioxidant activity. The hydroxy group that can be found in the leaf extract is responsible for facilitating the free radicals scavenging. So we are going to the last chapter, which is chapter 3. 3.1 Chemical and apparatus. In this final year project, a limited number of chemical and apparatus will be employed. Green mariposa Christia verpetolainis 1.9 Natrium titanium trichloride, distilled water, and Malipo water are among the chemical and materials that will be used in the green synthesis of AgNPS. In this study, the apparatus that will be used are the beakers, which are vary in size: 50 ml, 100 ml, 250 ml. The measuring cylinder: 10 ml, 50 ml, 100 ml. The magnetic stirrers, spatula, glass rod, petri dishes, Whatman filter paper, 100 ml Teflon autoclave reactor, volumetric flask, Buchner funnel, and the sonicator electronic balance, and the calcination funnel. 3.2 Plant extract preparation. The green Mariposa Christia verpetulanis leaf will be collected directly from plants. This leaf will be washed with distilled water for 4 or 5 minutes and dried under shade for 2 days. 10 grams of air-dried leaf will be taken in the beaker and will be extracted with 100 ml distilled water at 90 degrees for 2 hours heating. 0.21 Phytochemical test. For phytochemical tests, there are a few tests that can be done to the test for the presence of bioactive compound, which is the AgNPS. For alkaloid testing, when the 1% HCl, the plant extract, is mixed, warmed and filtered, the filtered then will be used for the next test. For the Meyer's test, the Meyer's reagent, which is a compound of mercuric chloride mixed with the potassium iodide and water. The filtrate was then be tested. The presence of alkaloid from the previous test will be then specified by the information of the yellow colored precipitate. The other test is the alkaline reagent test. For this test, the plant extract will be treated by using about 2 to 3 drops of sodium hydroxide. An aqueous yellow color will form that also indicated the presence of the flavonoids. If some drop of sulfuric acid were added, the yellow color will be changed to colorless. The last part is 3.3 Green synthesis of AgNPS. For the green synthesis of AgNPS, a 250 ml aluminium flask were used along with a 1 mm silver nitrate AgNO3. Aqueous solution was produced to produce AgNPS. In a nutshell, 10 ml of aloe vera leaf extract was mixed with 90 ml of aqueous solution of 1 mm silver nitrate and incubated at room temperature overnight in the dark. The color change from the colorless to colloidal brownish yellow confirmed the complete reaction of AgNO3 to Ag plus ion. After that, the colloidal mixture was bottled and appropriately kept for the future use. Spectrophotometric analysis confirmed the production of AgNPS.